Hello, everyone. Thank you for joining. We would be starting the session in just 5 more minutes. We are waiting for others to join as well. Hello, everyone. I would like to welcome you all for the webinar today on a very exciting topic, that is Space Omics, that is dedicated to the study of omics data to study molecular mechanisms affected by the space travel and critical for astronaut health as well. So, as we have a lot of new participants joining us today, I would like to first of all introduce what Space Omics is all about, and then we listen to the mentors discussing about the various research project opportunities in the domain itself. So, astrobiology is a science which addresses the origin of life on Earth and potential for life elsewhere in the universe as well. Space flight is one of the most extreme conditions that has been encountered by humans. And advances in omics methodology such as genomics, transcriptomics, proteomics, and metabolomics have revealed that unique differences exist between individuals. These differences can be amplified in extreme conditions such as spaceflight. A better understanding of individual differences may allow us to develop personalized countermeasure packages that optimize the safety and performance of each astronaut. Radiation, microgravity, altered atmospheric gas composition, isolation, and diet changes are just some of the known stressors affecting humans in the space environment. With extended mission durations and distances outside of the Earth's orbit, these factors are expected to increase. There are several adverse human health effects associated with space flight, including bone demineralization, skeletal muscle atrophy, cardiovascular deconditioning, vestibular control, immune system suppression, and neuroocular ailments as well. So, it is critical to understand how spaceflight factors affect human health in order to develop space safe and effective countermeasures. The NASA Twin study catalyzed the need for more comprehensive consortium based approaches to study the long term effects of spaceflight on humans. Here, 9 research groups studied a single data type in detail, whilst another group performed multi omic synthesis to determine the whole body changes. The study identified alterations in numerous data types, including telomere length, gene regulation, gut microbiome composition, body weight, carotid artery dimensions, and serum metabolite profiles as well. So, before we start with the talk, I would like to introduce us. We are a US based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy to use analytical tools, and our mission here is to make bioinformatics more accessible. Omixlogic research based training programs are a result of close collaboration between Pine Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. Omixlogic is an international program that is running in 5 different regions with over 10,000 users around the world. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and leverage our online training resources as well, adapting them to the needs of students and researchers around the world. My name is Sonalika Ray, and I'm the Omixlogic Community Manager. I'm responsible for daily interaction with thousands of users that are a part of our bioinformatics community managed by Pine Biotech. Our training is designed to allow participants to apply methods to curated and new data sets and develop independent projects that showcase what has been learned in the context of a personal portfolio of projects. As a result, anyone starting on this journey in bioinformatics can start developing a project of their own. So together with our existing modules for which you will gain access to the Omixlogic portal where you can try and explore these modules, exercises in pipeline building, coding and various methods of analysis using resources from peer-reviewed publications. Now, I would like to pass on the stage to Shubhrajit Barwa and Dilara Dickin to talk about their astrobiology projects and experience as well. Shubhrajit Barwa is an MS in molecular biology and biotechnology at the International SCAMP Laboratory, ITMO University, St. Petersburg, Russia. He is a graduate of Red School 2021 LaTeX, French Exobiology Society. He is an astrobiology educator at Art of Inquiry, where he works closely with Julia Brodsky, former NASA ISS astronaut trainer. He is also a volunteer at Mars on Earth project, where he works on Martian projects. He is a Russian government open door scholar and a President's Scholarship awardee as well. He was also one of the top 10 students globally selected for SGAC Space Health Risk Symposium London 2021. We are pleased to have you with us, Shubhrajit. Over to you.
Thank you so much, Sonalika, for that wonderful presentation. And hello to everyone who has joined us today. It's lovely to see you all here. And as Sonalika very accurately mentioned, that we are going to indulge more into this wonderful topic of astrobiology or space omics. And I'm pretty much sure that every one of you who is present here today is interested and enthusiastic, and you're well aware of the various kinds of research going on around the world. There are also numerous opportunities which are early career as well as for the more experienced ones be it a postdoctoral fellow or a scientist position available for various people all across the globe to grab this opportunity. To help people do that and announce yourself in the domain of astrobiology, we at Pine Biotech have organized this particular opportunity for all of you, where every one of you can get an opportunity to indulge in your own independent as well as team projects, where you would also receive mentor support, one on one guidance, and available course materials, which would help you to learn all along the way as you go. And if you have joined us for the first time, there was another webinar which was done. And in that webinar, me and my colleagues have discussed in detail about what are the topics that we are going to cover, what is astrobiology all about, what is our focus, and what is our aims. And in today's webinar, which is more of like a discussion, we would be discussing about all the project ideas that you guys have and what are your aspirations. We would also discuss about potential teams. You guys can form teams amongst yourself, those who are already here, or if you have a research lab or a team who would be interested, you can also invite them. And today we will be discussing about where to find the data sets, what the data looks like, and how can you start your own project just by sitting at your home. This is a pandemic hit era where it's very difficult to get hold of a laboratory to go to the fields and do the research work. Same kind of problem everyone is facing all around the world, but we can't stop, right? We have to go on doing our own projects and coming up with a publication, participating in conferences. And therefore, this sort of a model where you can actually get involved in practical works right from your home remotely provides you an exciting opportunity to be a part of bigger consortiums and groups just by sitting at your home. So before I move on to the further discussions, I would like to show you guys about what the entire plan is, how it looks like, and what is the plan altogether. So I guess you guys can see my screen. So this is the homepage of the program, which is called Introduction to Space Omics. And this program is actually designed you to help you through your research project. Now, there are several steps in defining a project and getting it done. For example, it begins with finding a good data set, a data set which is worthy of looking deeper into, to curate it, to label it properly, and to make it ready. The second part is obviously the analysis part, the methodologies that you have to use, and how you're going to use it, what is the appropriate methods that you should apply for your expected results. Now, once you get the hold of the results, how do you understand if the results are a good result or a bad one? Or what do you biologically interpret from those data? And how can it correlate with whatever is happening or going on in the biological body? So these are the various challenges which many students face in today's world. Whatever background you belong and whatever your expertise might be, these are the problems or the challenges which most of the people face. And that's why we have designed this particular curriculum to help you understand what we are going to talk about and what is it all about. So to begin with, the first day would be focused towards the topic of astrobiology. What is it all about? What are the various subtypes? As you can see on my screen, we will be also discussing about the theories of origin of life and the various topics or debates currently going on in this particular domain. The next day, which is on October 25th, we would be diving deeper into the concept of origin of life, because I'm sure that many of you are interested in actually analyzing or understanding the origin of life better, and you want to start some projects of your own use in this particular aspect of origin of life. So we would give you some idea about how you can gather data, what sort of data should you look into, what are the concepts that you can analyze. Definitely we would be talking about extremophiles, because you all must agree that extremophiles are very, very important in the understanding of origin of life or not, as well as in the concept of hoping for life beyond Earth. And we would just not give you another lecture, but also talk about the data sets. How does it look like, where you can find them, and also give you certain project examples which uses ancient RNA reconstruction or protein remodeling, and what results have they got, and what can you guys do as a part of your project. And this is not something which is very rare. Like, many research groups are actually doing marvelous research in this particular domain. Then the third day would be focused more towards the other aspect of astrobiology, which is space flight travel. So, as you would agree that in the current era, with the commercial spaceflight agencies coming to the forefront, traveling to space has increased manifold. And with the increase in the number of missions to space, the risks of astronauts getting affected by the various factors of spaceflight is also increasing rapidly. For example, spaceflight is made up of various factors like microgravity, ionizing radiation, and isolation that has an effect on their brain. So, before we plan a mission to Mars or maybe even the moon, which are long term missions, it becomes mandatory to completely understand the effects of these conditions on the genome and the expression of genes, the gut microbiome of the astronauts. And we need to make sure that they won't suffer from any chronic illness like cancer when they are on that particular mission. To do that, it's the right time to actually get hold of these sort of data sets and start analyzing them. And believe me or not, there are various organizations in the world who are looking into this particular domain, and they want people to get involved in this sort of projects. To give you an example, there's one conference which is coming up next month, and it's focused towards space health and the peaceful use of space technologies. So you can understand by the title itself that people are all around the world are actually talking about this particular thing. So it's a best opportunity for us to get involved in these sorts of conferences and other opportunities by having a project of our own. So that's what we will be talking about. And on the fourth day, since Pine Biotech focuses on bioinformatics, how to utilize the various omics data to your project, we would be giving you a thorough explanation of all the omics levels, like genomics, transcriptomics, metagenomics, epigenomics, and also multi-omics integration. This omics levels were studied in the NASA twin study, as mentioned by Sanalika. But here's the benefit of doing it with us. First of all, you don't need to have any coding skills. Many students who are biologists like me have a problem with coding. That's a big hurdle, but here you don't need to code. Why? Because here you would get all the algorithms just in a point and click interface. So you don't need to know about any of these algorithms, what's behind that, what are the codes. You just have to see this particular user interface, which prompts you, helps you with every detail which pops up on the screen. And you just have to click, point, and click, and you can build the entire pipeline. I would be giving you other examples as well and show you how to do it. And excuse me. So the final day, that is the day 5, would be again focused towards projects, because that's the day when you guys need to come up or decide on your projects and finalize what are you going to work on from this particular day. So this day I would be giving you other examples of projects and walking you through my project, how I do it, how I did it, and what my project looks like. Even today, I would be showing you some examples. And on this day, we would discuss with you all your confusions or doubts. And from this particular day, you would get the opportunity to work on your own project, and you would be meeting the mentors twice a month. So you would be getting the support. You can show them all your results, consult with them, and that final target, of course, is to come up with a good result which you can publish if it is excellent. If not publishing just now, you can definitely take it to a conference as a poster or an oral presentation. So let me give you an example of the various projects that has been already done by the people who have participated in the previous programs. So as you can see here, this is my project here. And if I scroll down, you would see that various other people have submitted their projects, which they have completed with Pine. So you can see these research questions, and they are the ones which are going around in the world of biotech right now. So here you can see the in silico analysis of affected biological pathways in multiple sclerosis, transcriptomic analysis of CD4 plus T cells. So every project that has been done here has been either published in a peer-reviewed journal or has been taken to a conference, and several students have won several prizes, in fact. So let me walk you through one of the projects and show you how it looks like. So the project here, if you go through, you would find that it's a well-structured interface where you would find a brief overview. You would find details about the data sets. You can check it out. And these are all submitted to the Harvard repository. You can visit them and analyze them yourself. Then comes the abstract part, where you would explain your project in brief. As you guys know, the structure of a peer-reviewed journal paper, basically, it's similar. So once you submit your project in this particular manner over here, it gets very easy to take it to a publication or to take it to any conference, because everything that you need is ready. So it looks really interesting, right? So before we move on to you guys and discuss with you what are your interests, and I would also show you some techniques to analyze it and some pipelines that I have created. But before moving on to that, we have with us Dilara Dekan from Turkey. And she has been one of those participants about whom I was talking about. And she has already done a project in this particular domain of astrobiology, where she has analyzed the radiation threats and the effects of radiation, ionizing radiation, on the gut microbiome of astronauts. And she has also taken her project to a preprint publication, and I'm sure she is looking for submitting it into a peer-reviewed journal. So it's the best opportunity to have a look and learn from her, have a look at her project and learn from her how was her experience of the project, how did she utilize the platform of TBINFO in Pine, and what's her entire experience, because there's a similar kind of experience that you guys are also going to experience in just a few days. So hi Dilara, over to you now.
Hello, thank you for that introduction. And I will quickly share my screen. One second. I hope everyone can see clearly. So as Subhajit said, I'm Dilara, and I'm a former Omixlogic research fellow, and I'll be taking you through the process of conducting research on astroscience using using omics through the TBioInfo platform. First, I will tackle some key points in research, like how to come up with a hypothesis, where and how to find good data, and how to convince an audience that your research is relevant and important. Then I will take you through my research and show which data and graphs are best to present and which ones I chose. And we will even practice a pipeline practically. So the hypothesis could be the start of your research, or vice versa. For me, for the purposes of my research, I decided to start here. So while I was reading articles to find a topic that intrigued me, I came upon the gut-brain axis, and since my preferred field was astrobiology, I decided to study how the gut microbiome affects the nervous system of astronauts. My hypothesis was based on how high LET, linear energy transfer, ionizing radiation affected the nervous system of astronauts. My hypothesis was based on this type of radiation for several reasons, like the fact that it causes chemical reactions. I surmised that this type of radiation would have adverse effects on the human body and the gut microbiome. Now, on to finding a good data set. You can actually search for articles that are published in high impact factor and well-known journals, because they are very well peer reviewed, very rigorously peer reviewed, and you should also try to pick ones with high sample numbers, since you can get better results in machine learning techniques with huge numbers of samples. As you can see, mine was categorized based on the radiation and time, and I had 80 samples. And of course, you can search NCBI and some other platforms like JGI for microbiome data. And as for the pipeline, it is important to choose an appropriate pre-processing and processing method, since you have a lot of data that you have to filter through when you have huge amounts of data. And you can do this using the tBioInfo server. So if you would like, we can try a hands-on example, and then I can take you through which graphs I chose from those results. And then we can continue like that. So I guess the link is already in the chat. So if you could quickly go there, and you will come to this page where there are numerous different areas of analysis. And from my type of data, I chose Amplicon 16S, 18S because of the type of data that was in the article was 16S rRNA data. So here you can see a demo. And if you are following with me, we can, here you can see as well, pre-processing. For this demo, there isn't pre-processing, but for my data there was, which helps remove primers, etc. And here we are trying all of them. I hope everyone can see, because I can't see the chat right now. So if you have any problems, please let me know. And what is great is the Data2 pipeline here and the phyloseq pipeline, you can run them both together at the same time. And then here you can see, I will pick this one, the Data2 and phyloseq plots PDF. And if you go there, here, you can see the abundance plot here, a heat map showing the differences in the abundances, a phylogenetic tree showing both the abundances as well here, the Shannon index that I will be explaining in detail. And here we have NMDS plots. So you can go through those in your free time if you would like. And now I'll go back to my data specifically and show you. So basically, my NGS data, which is the 16S rRNA data, was collected from feces samples of mice exposed to different types, to 4 different doses of radiation. And these samples were collected at 10 days and 30 days post-radiation. So from these results, I decided to pick the NMDS plot, which is a non-metric multidimensional analysis plot, which is a data reduction technique, which means that you can input high amounts of data, and it will, the machine learning algorithm will filter through this data, and it will only give you the points which have great variance and have a great effect on the data. So you can see here the differences between the groups, and at 10 days post-radiation, that group is slightly more separated than the others. So the next output that I chose was the Shannon index, since it gives us a general idea about the richness and evenness of bacteria in the samples. It is a great plot because it summarizes everything, the alpha diversity, the beta diversity of the microbiome. And here you can see that for lower doses of radiation, like 0.1 and 0.25 GY, the diversity decreases more compared to the control group. So Next, I decided to use the phylogenetic tree, and this one has a bonus because we can see the abundances based on the categories as well. As you can see, the OTUs that have a great effect as here, the algorithm filtered those out for us. So OTU 3, 4, and 5 are the ones that I'll be focusing on in the next step. And here you can also see that for 0.1 GY at 10 days, it has the greatest decrease in abundance compared to 0 GY. To corroborate the results, I also conducted a t-test and an f-test on Excel. And using the tBioInfo server again, in the utility section, you can even generate heat maps. And that's why it's really great using this platform, because I didn't know coding very well, and I can't say I'm very efficient at it yet. So you don't need to know these things. You can directly start generating graphs without knowing how to code using this platform. So I generated this heat map, and as you can see, all the graphs that I use are the graphs that summarize large sums of data, which is helpful when explaining a new concept to an audience. Here you can see the difference in abundances for OTU 3, 4, 5, and 8. They are significantly decreased between 0.1 and 0 GY. I then decided to search the KEGG database for these 4 OTUs because I didn't have names exactly. So they corresponded to Lactobacillus and Bifidobacterium genus, and I found out that these have protein coding genes in the GABA pathway. GABA is gamma-aminobutyric acid, and it is a chief neurotransmitter in the central nervous system and causes depression and anxiety when in low doses. So we can conclude that at lower doses of radiation, especially 0.1 GY collected at 10 days post-radiation, the abundances of these 4 types of bacteria decrease the most. And since they produce GABA, we can say that it has a correlation to the depression and anxiety that astronauts suffer during long-term flights. And that my initial hypothesis about the gut-brain axis is also correct. Of course, further research is required, but this is a good precursor study, and probiotics can help alleviate some of the symptoms of these astronauts. So what did I learn personally during this project submission and while conducting this research? First of all, I learned how to sort through good data and find good quality articles. And you become more efficient at creating a narrative so that your readers and listeners can understand your research. The more you write, the better you get at it. And especially referring to your mentors during this stage is great, because they will help you throughout the process, and you can efficiently estimate the quality of the data as well. And then you will come out also knowing which graphs are best to recapitulate your data and how to summarize your research. And overall, if you are rigorous during this stage, you can also learn how to conduct scientific data in an excellent manner. So here are my references, and if you have any questions, feel free to ask me. You can inquire anytime. And thank you for listening to me. I'll pass it back to Subhajit.
Thank you so much, Dilara. It was an amazing presentation. I'm sure that all of you would agree that the steps that she had explained, the manner in which she could start her journey from a data set which she got from the publicly available domain, like NCBI, to actually get some good data results which are biologically interpretable and has some meaning, is what we all want to do it. But as you would also notice that there are certain terms, there are certain concepts or certain graphs which you might need some help to interpret. And at this moment, you might not be aware of all those terminologies as such. And that is one of the reasons why we have organized or designed this particular course, where we would be helping you to understand all those concepts before you start working on your own project. So by the end of this particular program, you would have a clear understanding of all the terms that we have used. You would have a clear understanding of the what's a pipeline and what kind of a strategy you need to follow to get hold of your results. At the same time, during the course of your project, you could also get help from mentors, you would get their advices, you would get your consultations. So you would be able to consult the mentors, look for answers, and get guidance from them as you go along. It's not that we would leave you after the program is over to explore on your own. Definitely, you would have an opportunity to work independently, but also you would get the support. So before I move on to some other project examples and show you how the entire thing works from scratch, I would love to know what are your interests. So it would be great if you head down to the chat section and write down as vaguely as possible what's your interest of research, or what's the absolute field of astrobiology which excites you the most. You can also write it like, I'm interested in origin of life, I'm interested in exoplanet hunting, I'm interested in space health. So it's completely up to you, and I would love to see some responses in the chat right now about what particular aspect of astrobiology interests you and what would you love to indulge more upon. You can also write down what are your other interests in general, or what are you currently studying, your degree, your level of education, and maybe I would be able to guide you better about what you might select as your project. So, guys, feel free to write down in the chat whatever comes to your mind. I won't be judging you. I would just indulge in a conversation and show you that what's the area where you can start and how far you are from your own project. Okay, I'm waiting for your responses. You can just write it in short, no need to explain it long. And I hope that you are able to hear me in the first place. And if I don't get any responses right now, I would like to pick you up and ask you about your interests one by one. Would you prefer that? Okay. So here I see one response from Ruth. So she is interested in tissue regeneration in space. So Ruth, you might be studying about tissue regeneration, which is done on Earth. Like you might have done some literature review, or you might be looking into this particular aspect, and you know that what are the various signatures that people study and what are the various challenges. Now you are interested to study that in how tissue regeneration happens on space, because I'm sure that you have this particular idea that how regenerated tissue would be utilized to help the astronauts when they're on a long-term mission, to help them heal, to help them uh, maybe do some surgery or patch. So that's a really, really cool project that you have. Yeah, surgery in space, right? So I got you, right? So that's a very interesting project. And then, so the first thing that you need to do in order to start or get started is find a good data set, to look for data sets. And that's something which I would show you a little bit today, as well as in depth during the course, during the program. We would guide you. And also, once the program is over, we would also guide you to get hold of a good data set for your own, a data set which is good, free from noise, and also well, well classified and categorized for the particular project. So I also see Zahra about asking about the bacteria in space. So there's two things which comes to my mind, and I would require a clarification from you. First one, are you asking for the bacteria which might exist on other planets or other places about the International Space Station? Or you mean the bacterium which we carry or the astronauts carry with themselves in their gut microbiome, in their gut, to space, and what happens to those bacteria? You see, there's two really interesting, equally interesting aspects of this particular question. And it would be interesting to know which one you are more interested about. I can also add in a third, which would be focused more about um, how a person or an astronaut might respond to bacterium in space. So we might assume that immune system might be affected. And I will show you some examples of some results of some data which does hint at the weakening of immune system when one travels to space. So how does he respond to bacterium? And if he gets an infection in space, then how can he combat with it? And what are the ways in which we can address that? So Zahra means gut microbiome. And that is similar to what Dilara had also focused on. So when you go to space, there's various factors which affects you. There's microgravity, there's ionizing radiation. The astronauts survive on a different kind of a diet, which is pre-processed. So now this particular factors have their own kind of effect or a combined effect on the gut microbiome. And as we all know, that gut microbiome plays an important role in many things, including immunity. It has a connection with the gut-brain axis, just like Dilara mentioned. So it would also have effects on your central nervous system, and maybe that can cause some neurodegeneration, which would be harmful. So what are the ways in which we can actually address those issues would be interesting to look into. And therefore, your entire topic is very relevant and interesting. I also see Ilya, he has mentioned that the T cell function is impacted. You're absolutely right. And microbes are also more virulent. The microbes activity gets more virulent and it affects more. So I think you can look much more in deeper in this particular field, because um, T cell has various functions. T cell has various categories, as you might know. There's T helper cells, there are cytotoxic T cells. Now, it would be interesting to know what factors affect them and what are the pathways which are affected as a result of the T cell dysregulations, and what are the ways which we can address them. Can we identify some particular targets which can be addressed or targeted with some drugs, and therefore make the future astronauts well prepared or equipped to actually combat all of these issues? So here are certain very interesting project ideas already. Like even before the course has started and before I have introduced you to the various interesting aspects, there's already so many interesting project ideas. So this is something which I was actually talking about, and it would be very interesting to know more. So all of you guys, for those who are here in the audience, keep typing, keep writing, and even if you don't have a topic, it's absolutely fine. There's nothing to worry about, because we have certain project ideas that I would like to share with you. You can also join me in my team, and I would love to work with you, and we can together explore the various secrets which are hidden. So now I would like to show you one particular example which I am looking into right now, and I would show you from scratch how I proceeded and how I went on. So here's my browser. So I wanted to analyze what happens to the immune system of the astronauts when they go to space. But you would agree that human generated data is limited, but that's okay. So where to look for data? NCBI is one very good place to look for data. The other one, for specifically designed for all of astrobiological work, is NASA Gene Lab. So here I'm going, and I will also give you the link in the chat so you all can visit it yourself as well. So NASA Gene Labs is an open source data repository where the data from each and every space missions, which are in collaboration with this particular organization, have submitted. So you can see here various transcriptomics data, various genomics, proteomics, various other processed data. You can also get a hang of all the publications that has been done. So I went to this particular repository. I went to the data repository section, and I started to look into various organisms. So here you can see there's this particular study which talks about the effect of microgravity on epigenetic modifications of Synerheptitis elegans and stuffs. I focused my study on mouse, so I went and looked for mus musculus. So here you can see mouse. I use a filter mouse to get all the mouse data, and I come across various mouse data which has been studied. I can also filter the various assay types, for example, DNA methylation profiling or genome sequencing, transcriptome profiling. So these are the various terms which I would be explaining you during the course of the program. And by the end of it, you would get a hang of all what it is if you already aren't aware of it. I am sure many of you already know about it. So here you can see murine T cell receptor beta repertoire sequencing. So someone, I guess Ilya was talking about T cell receptors. So here you can see there is data for T cell receptor sequences. So you can analyze them and get involved more. So let me go to the particular data set that I used, and the accession number for it is GLDS288. So it is the transcriptome analysis of mouse spleen. If you open this particular site, you would get the details of the mission in a very well structured manner. You would get all the protocols that were done and what is the various aspects of this data, how would you define them, were the metadata about the data sets, what were the conditions basically, what were the sexes, what were the age of them, and more. So I will show you how it looks like when you download this data set and load it onto Excel. So we will be using Excel a lot, so get ready to use Excel profoundly. And don't worry, we would also cover it in the program, and also you would find some free materials on the Omixlogic platform, which talks about how to use Excel and how you can get a hold of everything on Excel. All right, so here you can see that there were 3 categories of samples. The first, which were cultured in a centrifuge, but in 1 gravity, like the normal gravity that we are used to. And this is the main control that we have. So centrifuge is done just to replicate the stress on the organism. Then we have the ground control, which does nothing to the mouse mouse models. They are just normally on the ground. And the third one, where the actual mice which went to space aboard SpaceX Dragon capsule, and they spent around 35 days aboard the International Space Station, and then they were brought to Earth. So now you have various genes which you can identify here with their gene IDs, but we want to look at the signatures. We want to identify which genes might be affected and what does that show, or what does that mean to us. So that's where the platform of tBioInfo comes handy. If you look at my pipeline here, which I used, so I used a pipeline which is known as a differential gene expression pipeline. And here you can see all the algorithms that are involved in this particular pipeline. And I just use 3 of them. So when you click on start, you go to the differential expression algorithm, which is DSeq2, enrichment analysis, gene set enrichment analysis, and that's the end. And once this pipeline ran for some amount of time, it gave me various results. So here are all the results that it generated. That's a lot, but it's also very interesting to look into all of these results. So once I downloaded all these results here, you can see now we can actually open some of these plots and diagrams and have a look at what it means and what are the various interesting pathways that we can look into. First of all, we get some volcano plots. This is known as a volcano plot. It basically shows whether there are certain genes which are highly upregulated or downregulated, and amongst them, which are the most interesting ones. So you can see some of the names of genes here. So you have the evidence that, yeah, something is going wrong when these mice visit space. Next up, you would see this particular plot, which is a graph, and it shows that which genes are co-expressed in various pathways. Here are various pathways, and some genes are involved in more than one pathways. And in this particular, amongst these particular pathways, in which pathway is this particular gene highly expressed or low expressed, downregulated or upregulated? And you can identify them and you can do further analysis. Also, there's this particular plot which shows which pathways might be affected and upregulated or downregulated. And you would find some very interesting pathways like activation of immune response, myeloid cell homeostasis, response to bacterium, erythrocyte homeostasis, complement activation, the complement pathway activation, iron-ion homeostasis, phagocytosis, recognition, and more. So you can understand that with the help of a few clicks only, you could get insight into so many different biological pathways which are affected, how they are affected, what's the amount by which they are affected. And from here, you can go on and study their interactions as well with the help of these networks, how these genes are connected with each other, what networks do they form, and amongst this particular network, what are the genes that are interesting to look into. So there are various pathways that you can look into. Some of these networks are very complex, but it's still very much informative. So with the help of this particular data and with the help of the other data that are generated, I would also like to show you what we can do it with the differentially expressed genes. So this is how the differential gene expression actual data looks like on Excel. And if I do a count here, so there are currently 17,907 genes, but not all of them are interesting to me. Why? Because not all of them might be significant. So how to set a filter for significance? So we have to set parameters in the log2 fold change, and let's set it between 2 and -2. So it should be greater than or equal to -2 and less than or equal to 2, so that a log change of 2 would be a significant one. We can also set some filters for p-value and p-adjusted value. It stands for the Benjaminian-Hodgman threshold value. We usually set it to 0.05, and we can also finally apply one sorting and sort it from lowest to highest. So now, for example, let's look at the first gene that it shows. By the way, remember we had 17,907 genes to begin with, but now we just have 40 genes. So now our data set is much more refined and polished. We just have 40 significantly affected genes, which would be interesting to look into and see how are they getting affected and what does that mean. So the first one, the first gene here has some crazy ID, but don't worry, once you run it in the platform, on the Pine Biotech platform, you would get the name and the symbol of the platform, and you can guess what it does. It's hemoglobin beta added T chain. So you get a hold of what this particular gene is and what it might do. So you can also go and explore more in NCBI to learn more about them. So once you select this particular genes and you would like to do a plot right here to see how are they affected. So here you go. So you see the first 3 samples, as you might see here, are microgravity, or the samples or the genes in the mouse which went to space, as compared to the other 3 which were on Earth in a centrifuge. So clearly you would see that the first 3 samples have suffered a downregulation of this particular gene, and they are suppressed, whereas the other 3 are high, like maybe they're expressing normally without being affected, that we don't know yet. But what's interesting is that these 3 have suffered a downregulation. Now, what does this downregulation mean? So that would be interesting to look into further, that what does the downregulation of this particular gene have the effect on the phenotype of the organism? Does it cause some pathways to go wrong or something like that? And the pathway that I use, the pipeline that I used also generates something known as pathway enrichment analysis. And this result also gives us the names of the pathways which are affected. So here you can see PI3K, AKT signaling pathway, calcium signaling pathway, the spliceosome, axon guidance. And immediately when I look into these names, what I'm reminded of is molecular neuroscience, because all of these pathways are important in neuronal activity. But we used spleen tissue for our sequencing. And now that we are getting some hint at the pathways that are affected in the spleen, they are affecting both the immune system as well as the nervous system or the central nervous system. Now, it would be interesting to look further that how does this particular pathways going wrong in the spleen can have an effect on the brain. And if it is so, then maybe the people who go on a space travel actually might suffer from some form of neurodegeneration in the future. They might have or develop some neurodegenerative diseases like Alzheimer's disease or schizophrenia or something like that. So this is the entire thing where you would like to reach as the end goal of your project. And if you actually reach into something of this sort, definitely you would be able to publish it or take it to a conference and talk more about it and discuss with other experts about what does these results mean and what are the effects of these results. So that's what I showed you. And now I would like to ask you, or would like to give you the floor to ask any questions that you might have to me about this particular project or about the entire program. And this is something which we want to achieve with you guys as well. And when you enroll for the program, we would first guide you about all these terms. Let me show you one more thing as well. When you go to the Omixlogic platform, you would find that there is an option called courses. And if you go to the courses section, here you would also find, see, various courses which are free, like Introduction to Bioinformatics and other stuffs. Specifically, you can also go to transcriptomics, for example, from this tabs here, because I was talking about transcriptomics. You would be interested into looking at how or what are the various projects and the various courses that are available on the platform. I would also show you something else. Just give me one moment to find it out. So here's one very interesting and free course that is available on the platform itself. It's called the Bytes and Molecules. So I would recommend you to go through this for sure. You would understand what we are talking about. You would understand what is the DNA, what is DNA, RNA, and what are we looking into it, how are we looking into it. And finally, in the end, you would also find the last course, that is Space Omics and Astronaut Health. So this is a course which has been specifically designed for this particular course, where if I show you, you would learn a lot about this particular domain. You would learn a lot about what's going on, what's suggested by NASA, what we mean by microgravity, what are the various methods of simulating microgravity, and what are the various data repositories. Like I was showing you NASA Gene Labs, you can see and go through how NASA Gene Labs look like and what is it. So there's one more thing which I would like to show you is the transcriptomics course. If you go here, you see this is a very nice and classy explanation of what transcriptomics is, and it basically explains everything that I did in my project that I was talking to you and showing you. You see there's next generation sequencing explained here, what does the data look like, and what are the various algorithms that you might use here to analyze your own data set. Similarly, you would also find courses about genomics, about metagenomics. So whatever your interest might be, whatever is it that you are interested in, you can definitely go through these courses on your own without us intervening at all, and look up on your own, understand about what is it and how you can apply or implement these things in your own project. So here also you can see RNA-seq, the pipeline has been explained. You're also getting a practical to do the RNA-seq practical here. And also then there's other statistical tests like the t-test. You will also learn about differential gene expression. You see, I was showing you certain results of this sort, this particular graph or plot. So it's explained in this particular course about how to interpret it, what it is, how it looks like. There's other machine learning algorithms as well, like the unsupervised machine learning algorithm of PCA and more. So this particular course is not free. As you can see, there's a star here, but to all the participants who register for the program, you would get access to all these particular courses and more about the payment ways and how you can do it and what are the benefits of it, what are the various labs of uh, subscription that you can avail would be explained by Sonalika. So now I would pass on the platform to Sonalika to explain and guide you through all the payment stuffs and what are the benefits and what are the courses that you can access which are otherwise paid, but now you can access it because you have the subscription for this particular course. And meanwhile, if you guys have any question out of the blue, whatever it might be, doesn't matter, put them in the chat session and I would be happy to address them. So over to you, Sonalika.
Thank you so much, Shubhrajit, for such a wonderful presentation of the project and whatever you worked on, and also the way you explained your results was amazing. And thank you so much, Dilara, as well, for your explanation of the project. And now, for those who have not registered themselves for the program but are interested to do so, so they can find the link to the program page in the chat box that has just been shared by Ojasvi. So here on the program page, you would be able to find all the details of the sessions which are scheduled for the program. The calendar shows all the dates on which the sessions are scheduled and also the topics that are going to be covered for the program. Now here on the program page itself, you would be able to find the Zoom links sessions once you are a part of this page. And also once the session is over, you would find the recording of that particular session on the page itself, and you can just go through the recording at your own pace. Now, many a times what happens, the person is not available at the live session time duration. So for you, it would be very easy to just go through the recording and take up doubts in the common Slack channel that we use for communication. And to register yourself for the program on the registration page itself, you would find 3 levels: beginner, intermediate, and advanced. Now, these 3 levels are actually differentiated just based on the facilities that are being provided for the program, right? So here in the intermediate level, you have the access to one-on-one mentor support where you can have one live session with your mentor, or like a one-on-one meeting for 60 minutes. And also then we have the advanced license where you will get extra time to work on a project. One-on-one mentor support would definitely be there, and also the license to the server would be the research license, whereas in case of the beginner and the intermediate ones, it would be an educational license. So that is the difference between 3 levels. Whichever level you would like to enroll yourself, you can just select this the option and just proceed with the payment gateway. Now, there is one thing. There are many countries or areas where the international banking is not active for the participant. So you also have alternate options, and you can just mail us at marketing@pine.bio, and we can just share out the alternate option details with you. It would be a PayPal link, and also for Indians, you can use the bank transfer option directly, or the GPay or the other modes of internet payment. And also on the program page itself, you would be able to find the program course and project libraries to which you will have the access on the portal. Now, these are the asynchronous course materials that you are supposed to finish up as a part of the program itself. So to begin with, what we'll suggest, as well also the people who have already a part, have joined the program page and are a part of the program already, for them we had recommended to just go through the introduction to space omics lesson in the bytes and molecules course, and also go ahead with the course on introduction to bioinformatics, so that if you are from a non-bioinformatics background, you get some insights and some understanding of what the role of bioinformatics is in translational healthcare. So this is the introductory lesson that I was just talking about, space omics and astronaut health, that you are supposed to just begin with and complete till the time we have our first session that is scheduled for 19th of October, right? And then you have the other courses as well that you are supposed to complete asynchronously as a part of the program. Now, once your courses are complete, the asynchronous ones, and also once your sessions are complete, you would be able to find the certificate on your profile. Now, when I talk about profile, first of all, it's very, very important that you have an updated profile. That is, you have an updated profile picture. Your name should be in title case because that's the same way your name would be highlighted on the certificates. So please don't write your name in caps or in small letters. It should be in title case. Also, there would be many people who might not want to begin with a specialized course on space omics now but would like to join later. So for them you can just explore the portal and start with the free courses first of all, and then go ahead with the basic subscription, so that gives you the access to the start lessons on the portal itself in the courses. So it's important for you to have an updated profile. And when you complete those courses, you will have your certificates in the profile section itself, and you can simply download your certificates from here. Also, it's important that you connect your social media account. And why it's important? Because we do feature people, participants based on their progress. So we would be just going through your progress here on the back end and monitor it. And we would definitely feature you seeing your progress, right? And also, it's important that you put a brief bio about yourself so we do know about you all. And those people who have already joined the program, please make sure that you have the Space Omics badge here under your profile tab. So having this badge gives you a sense that you are a part of the program page, and you will get the access to all the Zoom session links and also the recordings of the sessions that will be uploaded on the page, right? And here in the courses tab, you will be able to just look at the courses that you have started with and what you're working on right now, and then take up the certificate. So that was what I wanted to share with. And now if you have any doubts, please ask in the comments section so that we can just guide you through. And you can also mail us at the mail ID that I've given it. Also, we have an alternate mail ID that is marketing@omixlogic.com. And we would be happy to guide you through the process. Thank you so much, Shubhrajit and Dilara. It was great to have you both here, and we are looking forward to your sessions in the program. And also, I would like to thank you all for being a part of the webinar today. And we would be definitely looking forward to meet you all on 19th as well. Thank you, everyone. Have